Hello student, welcome to the lecture. In previous lecture we discussed the Metalloenzyme of molybdenum. Now in this lecture we are going to discuss the Metalloenzyme of Cobalt, that is vitamin B12. As we know that the vitamin B12 is the first organometallic compound naturally occurring. So here if you see the figure, in that the cobalt is in a center which is in +3 oxidation state, and here the ring is present in vitamin B12, that is a corine ring. The groups attached to these corine rings are a very complex structure. In that, depending the fifth position is fourth nitrogen are occupied in this corine ring, and fifth and sixth positions, actually the fifth position is occupied by the phosphate group, whereas the sixth position is occupied by the alkyl group. Alkyl means that there is any methyl, hydroxy, cyanide. These groups are attached with this are alkyl group. So here these are the structure. This is the structure of vitamin B12. Now let's discuss in detail the features of the characteristics of the vitamin B12. As we know that the vitamin B12 is a first naturally occurring organometallic compound. So the vitamin B12 naturally occurring organometallic compound vitamin B12 is also known as cobalamin. Is water soluble involved in metabolism of every cell of the human body. It is a cofactor of 8 it is vitamin of it is one of 8 beta vitamins. Vitamin B12 is a cofactor. In DNA synthesis and both in fatty acid and amino acid metabolism. So these are the general characteristics of vitamin B12. As we know that in vitamin B12, cobalt is in center atom which is in +3 oxidation state here, whereas this is a corine ring, and this corine likewise porphyrin ring. The vitamin B12 contains the corine ring, in that the one ring is in flat manner. So here it easily transfers energy, or it will get excited from singlet to triplet state. And here, if suppose R is CH3, cyanide, OH and 5 So these are the all general groups are present. According to these groups, the vitamin B12 is again nomenclatured as, if suppose there is a methyl group is present, then this is a, if suppose there is a methyl group, if R is methyl group, then this is a methyl cobalamine. Whereas R is cyanide, then there is a cyanocobalamine. If R is OH, then hydroxocobalamine. So these are the all nomenclature according to which group is present at the position R. So these are the general characteristics of vitamin B12. Vitamin B12 helps in maturation of red blood cells, in bone marrow, so this is one of the important function of vitamin B12. So these are the general characteristics of vitamin B12. Let's discuss the vitamin B12 and its model compounds. The model compound of vitamin B12, as we know that the vitamin B12 play a very important role in red blood cells in bone marrow. So here we are going to discuss the vitamin B12 and model compounds. The model compounds of vitamin B12 is a cobaloxime. Cobaloxime is similar to with the glycoxime. So their functioning, the model compounds of vitamin B12 means that their functioning and reactions are very much similar to the vitamin B12. So let's discuss the features or the characteristics of the cobaloxime. Cobaloxime is a model compound of vitamin B12. As we see in figure, there is a cobalt is at the center and 4 nitrogen like vitamin B12 is also present. The chlorine ring with this 4 nitrogen, and the 5th and 6th positions are here occupied by the two group, that is R, then it is an alkyl group, and second that is the beta B, means it will be the separate notation for R and B. Let's see. In cobaloxime, R is organic group, it is sigma bonded to cobalt. For example, alkynes and B is any neutral ligand. Is a neutral ligand. Neutral axial base ligand. It is situated at axial position, so that's why it is an axial base ligand. Trans to COC bond. That is COC bond, means here there is an alkyl group, and this position is trans to this beta, means there is an active neutral axial ligand. So this is COC bond. For example, pyridine, H2O, pyridine and H2O. Next one, the equatorial dioxins model. The chlorine ring of B12 coenzyme and crystallography data available on cobalamines are suggested that the structural effect of changing axial group are similar to those found in cobalamines. So, as we know that the cobalamoximes are very much resembles, or the reactions are stimulated by cobalamoxime is very much similar to the vitamin B12. So that's why the cobalamoxime is a model of vitamin B12. Now let's discuss the next characteristics. Cobalamoximes is also helps or function in to determine the properties and action of vitamins, as it is model compound of vitamin. So here the B group is, as we know that the B group is in water or pyridine, and this is an alkyl group because the reactions of cobalamoxime the reaction is that cobalamoxime is slowly decomposed by acid and bases with acids. The product of decomposition are dimethylenzyme, cobalt salt, pyridine, etc. Actually, cobalamoxime does not react with hydrogen gas. So the second reaction is that cobalamoxime is not reacted with hydrogen gas, as the reduction of cobalamoxime at near pH 7 is reduced to upper second centers at higher pH again higher pH, the Cobalt is further reduced to copper first oxidation state, cobalt first center. Vitamin B12 and its model compounds.